Everyone, welcome to our first video about the scientific method. Today we're going to just talk through some of the steps of the scientific method and how it works in a science classroom. So the very first step of the scientific method starts when a scientist makes an observation about the world around him or her. Next, they might ask a question and conduct research. Then they might form a hypothesis. And then next, they're going to test it. A test of the hypothesis is also known as an experiment. After the experiment or towards the end, they're going to collect and record data. And finally, they're going to make conclusions. But that's not really the last step. The actual last step of the scientific method is to replicate and retest over and over and over and over again to try to continue showing that your results either match or do not match your hypothesis. So the scientific method kind of loops. Once you've made a conclusion, you can think about your hypothesis and whether or not it was supported or not. If your hypothesis was not supported, you might make a new hypothesis, do a new experiment, and come to a new conclusion. Then, if your conclusion was supported, you might test again and again and again, reproducing the experiment until eventually it becomes a theory and you can go on to do further experimentation based on that theory. Observations here, they're gathered through your senses like smell, taste, touch, sound, and sight. Scientists can notice something that's going on around them, or whether that's what in the natural world, like you can see in that picture with the child observing the butterfly, or whether it's in the lab where this young scientist is observing a chemical reaction. You can make observations of any type, though be careful in the lab setting, you're most likely not going to be making too many observations of taste. Next, after you've made an observation, you will do some research. You want to use a scientific journal to do your research. This could be something like Nature or Science, and there are many different types of scientific journals that have been peer reviewed, which means that scientists have had other scientists read their work and make sure that it is legitimate. Here's the thing, you don't necessarily want to just use some random newspaper article or a tabloid or a social media post as your research. That is not legitimate research. You need to have written scientific journals as part of your research if you are going to begin making hypotheses. When you make hypothesis, you are suggesting a solution or an educated guess about what might happen if you do an experiment. Your hypothesis must be something that you can test, or basically something that you can conduct an experiment with. They're usually written in an if-then statement. So if you water plants with soda, then they will not grow as well as if you water them with water. They predict an outcome of an experiment. So once you've made a hypothesis, you are going to test it. And if you test it and your hypothesis continues to show the same results in an experiment, then eventually you're going to create a theory. A theory is basically a description of the world or a set of hypotheses that relate to one topic and have met observational experimental tests, which means that basically you've tested the same hypothesis or set of hypotheses over and over and over again, and now you have a theory. A law of nature is a theory or a group of theories combined that has been tested very, very, very extensively, and it seems to be true pretty much everywhere in the universe. So it becomes part of the main set of information that we know about a certain field. So in biology, we don't have many laws of nature. We actually have more theories like germ theory or cell theory in biology, but in chemistry, we actually have many laws like Charles's law and Boyle's law and Avogadro's law that tell us how different atoms and substances will act in certain conditions. So you will see that in the coming year or years. The difference kind of between these different terms, hypothesis is less absolute and less certain. It is developed from facts that scientists have researched, but it's kind of the least absolute of these three words: hypothesis, theory, and law. Theory is A little bit more absolute than hypothesis. Theory is something that's been tested and experimented on over and over again. And a law is more or less proven. It's the most absolute information. Typically, theories can change and be kind of overwritten or overridden, but laws do not. They've existed for a very long time and are proven to be true in many different settings. So, from most definite absolute to least definite absolute is going to be law, theory, hypothesis. An experiment is when you test your hypothesis, and experiments can be part of developing theories and laws, but they're basically procedures that you can do to find out whether your hypothesis was correct or incorrect, or in statistical terms, whether it's supported or not supported. This is just an example of a biology experiment that could take place. This is a chemistry experiment that involves distillation that could take place. Variables within an experiment. You might have heard of the IV and the DV. The IV stands for independent variable. That's the variable that you, the scientist, control. So I like to say I control the independent variable. They both start with I, so that can help you remember. The dependent variable is the thing that you're measuring. It's what you want to observe as the experimenter. Data comes from the dependent variable, or DM. Dependent variable is measured in the experiment. So IV is the one that you control. There might be different levels of that. So let's say you're doing an experiment where you're watering plants with different substances. Your independent variable might be what type of substance you're putting on the plant. You control that. So it could be water, soda, or milk, for example. You're controlling that. The dependent variable is what you're measuring. For example, in the plant experiment, you might be measuring how tall the plant grows. That's dependent for what is measured, DM. Mix and dry are two expressions that can help you understand which variables are on which axis when you're graphing information, when you're reporting your results of your lab. Mix is referring to the x-axis. That's the manipulated or independent variable, and it's on the x-axis. Dry stands for dependent or responding variable, the thing that you're measuring, and that goes on the y-axis. So independent variable goes down here on the x-axis, and dependent variable goes here on the y-axis. So in the plant example, your x-axis might have different areas for the different types of drinks you put on your plant, and the y-axis would measure how tall did the plant grow. How many variables would we purposefully change in an experiment? It's actually just one. So in the plant experiment, we're only changing the variable that is what type of liquid is poured on the plant. We're not changing anything else. We're not changing how much sunlight it gets. We're not changing how much of the liquid it gets. We're not changing what kind of soil it's in or anything else. We're just purposely changing one variable so that we can measure the impact that that had on, in this example, the plant. The key idea here is experimenters change one factor, the independent variable or IV, and they observe or measure what happens. What they're observing or measuring is the dependent variable or the DV. All right, because we only want to change one variable, all of the other factors or variables will be kept purposefully the same throughout the experiment. Like I was saying in the example before, if you're changing the liquid that you're watering plants with, everything else needs to remain the same. The amount of sunlight would be a constant. The amount of soil would be a constant. The type of soil would be a constant. The amount of liquid you're using to water it would be a constant. The species of plant that you're growing would be a constant. Everything else, except for that one thing that you're curious about, must stay the same. And those factors that you're keeping constant are called your constants. There's another thing within the scientific experimental design that is called the control group. Now, if you're going to make a claim that soda makes plants grow better than water, you have to basically be able to compare that soda back to water, which is considered the normal thing to water plants with. Control groups are the thing that's not being tested. It's kind of used for comparison. It's typically what would be the normal treatment. In order to have a valid experiment, you must have at least two groups, one control group, such as water, and one experimental group, like soda or milk to water a plant, and only one, keyword one, independent variable. You're only changing one variable within your experiment. Now, there could be multiple levels like milk, water, soda, dish soap, something like that. You could have multiple different levels of that independent variable, but you just have one independent variable, one thing that you're purposefully changing to see what happens. Data. Results can be collected and it can be shown in charts or tables or graphs, but results are either going to be qualitative, which it has to do with qualities or words, or it could be quantitative, which has to do with numbers. Once you have your results, you're going to make a conclusion. Your conclusion is going to be an answer to your hypothesis, whether your hypothesis was supported or not supported by the experimental results. Parts of your conclusion are going to be: what does your data mean? What about your data supports or does not support your hypothesis? And you might have any sources of error that could have impacted your experiment. And you can also discuss improvements for future experimenters. Because, like I said, you'll want to repeat and redo your experiment over and over and over again if you want to contribute to making a theory. When you retest, you're going to verify the results of your experiment. So you must retest it. Well, actually, you must not, but you have to publish your results so that other scientists might be able to retest the same experiment and see if that hypothesis will be supported over and over and over again, or if the hypothesis ends up not being supported. Science is durable yet open to change. What that means is that we have a strong set of scientific knowledge, but it's constantly being re examined and retested. And because of that, our knowledge can either become stronger in a certain area or it can change. For example, with germ theory, we used to think we had this thing called miasma theory where bad air caused diseases, but now we know that it's actually caused by germs like bacteria and viruses. So science is durable, yet there can be changes. Thank you so much for listening.